Okay, guys, last question here, number six. It's on the digestive system and breaking down food. And we've got a diagram. So make sure you could label any bits of this diagram. Specifically here, you're being asked to identify the stomach. A good tip is to trace the tube going from the mouth. And so if we go in the mouth at A, down the esophagus, the first organ we come to is the stomach. So we're going to tick C. And then that tube continues into small intestines, D, large intestine, and then leaves the body via the rectum and the anus. Organ B is the liver. So most people maybe would confuse that with the stomach, but if you follow the tube, you'll make sure you get your diagram right. What type of enzyme is produced in the stomach? So remember that proteins are digested in your stomach. We get protein from meat and fish and beans, and proteins are broken down by protease enzymes. The ones in your stomach prefer it to be acidic, and that's why, or one reason why your stomach has such a low pH. So on to question C, exactly that. The pH in the stomach is acidic. It's an acid. Your stomach makes hydrochloric acid. And the reason you can have here is either it's in order to kill bacteria that you swallow with your food, but also, like we just said, it provides the optimum, that means the best, the optimum conditions for protease enzyme to work. Part D, which organ produces bile? Fact to learn, bile is made in your liver. And how does it help? It makes fats break down into tiny droplets. So the reason tiny droplets are easier to digest is because there's a bigger surface area for the lipase enzyme to work on. The next part of this question is about food tests. It says a student tested different foods for the presence of protein, starch, and sugar. Draw one line. Remember our rule, for goodness sake, don't draw more than one because you won't get any marks. One line from each food molecule to the reagent to test. So there's only one correct set of answers here. Again, if you're certain, get one out of the way, and that gives you 50-50 on the other two. So starch is tested for with iodine solution. So we need to join starch up to iodine, and then I need to decide between the last two. You're just going to have to learn these. Protein are tested with Biuret reagent, and sugar is tested for with Benedict's solution. A safety precaution with Benedict's, if you were using any of these chemicals, you would wear goggles. So if you're ever unsure about safety precautions, wearing goggles is pretty much always right. So let's stick to that. H is our results. So again, we've learned the tests: starch with iodine, sugar with Benedict's, protein with Biuret, and then we need to know the colors. So Benedict's solution and Biuret solution are both blue to start with. The Benedict solution goes orangey red if sugar is present. The Biuret test goes purple if protein is present. So that's a good way to remember that. Biuret for protein, purple protein is the color we're looking for, and iodine is orangey brown to start with, and it goes blue-black in the presence of starch. So if we look at our results here, we've got Benedict's has gone red, Biuret has stayed blue, and the iodine has gone black. So those results are telling me that there are positive tests for Benedict's and iodine. It means that this food sample contains sugar, that's the Benedict's test, and starch, that's the iodine test, but doesn't contain protein. Last question. Starch molecules are not absorbed into the blood, and this is the whole point of your digestive system. It breaks big insoluble molecules into small soluble ones, so they can be absorbed. So if starch cannot be absorbed, if anything can't be absorbed, it's because its molecules are too large. Another way of saying that would be to say that the starch is insoluble.